Anyway, the first technique that we're going to talk about is called the polymerase chain reaction. Now, notice that the name here kind of gives away what this does. We know that polymerase is a enzyme that builds up DNA and RNA, right? There's an RNA polymerase, there's a DNA polymerase. So my guess, right, your guess would be that this laboratory technique probably results in the production of either DNA or RNA. This is abbreviated PCR. And I just was at Ms. Riley's class about an hour ago, picking up some lab equipment that she has. She lets me borrow some of her equipment. She actually has a PCR machine. And this machine, the reason why they're so expensive is because they run through different temperature cycles and they go back and forth to these different temperatures, and it's all monitored and regulated. So it's a very expensive machine, but if you take Biomed 2, aren't there 2 classes of Biomed 4? If you take Biomed 4, you will do PCR. Okay, anyway, what this is it amplifies. It's kind of a funny way to say it. All that they mean by amplifies is makes millions of copies. Okay, but you're gonna hear them say amplify. Makes millions of copies of trace amounts of DNA. All you have to have is just a teeny little bit of DNA, and you can run it through the PCR and get millions more copies. So it can be studied. So the examples would be like DNA analysis for oops, hang on, spelled that wrong. For evolutionary relationships, or you could use it for crime scene investigations, like if you're trying to match DNA to a suspect. Those are all practical reasons why we might want to copy the DNA. And here's the real reason, right? When you go, like when a CSI, investigator is trying to collect DNA from the crime scene, do they usually get a lot of DNA, or is it like just the teeniest little amounts, right? They always end up with just these teeny amounts, but so they don't usually get enough. Like maybe it's like a single hair, right, from the crime scene. They don't have enough DNA in that single hair to do a whole bunch of tests. So the very first thing they have to do is amplify or make millions of copies of the DNA so that they can do as many laboratory tests as they want later on with the DNA. So that's the reason that we have PCR. You make more copies so we can do more tests. Now, there's some ingredients that go into this. You have to have something called Taq polymerase. And this is a heat-tolerant version of DNA polymerase that is derived from thermophile bacteria. So there's these, or I guess I should say thermophilic bacteria. There's these bacteria that live in really hot places like, what do you call it? Geysers, and just like, I forget what those are called, but like Yellowstone has them. Those hot springs. Yeah, thank you. Hot springs. And there's bacteria that live there, and they've evolved and adapted to these really high heat conditions. And so therefore their proteins are tolerant to these high temperatures. And that's exactly what we need when we're doing this polymerase chain reaction, because the temperatures are high. So we need, if you were to use just a regular DNA polymerase, it would denature from the high temperatures. So we have to have a special polymerase. Okay, the other thing we need is we need primers. Primers are small segments of DNA that help get it started. And they bind to very specific locations. We also need a source DNA. That's the DNA sample, right? And then we need lots of DNA nucleotides because those are the building blocks. And if we're going to build new DNA, we need lots of nucleotides. And then the PCR machine, obviously. That's not a real organic ingredient. But in terms of the organic ingredients, we need the Taq polymerase, the primers, the source DNA, and lots of DNA nucleotides. Okay, so like I said, this is a cyclic thing. So the machine will repeat these cycles, and I think it goes through 20 cycles. Okay. But anyway, each cycle consists of these steps. The first step is called denaturation, which means that the heat will separate the DNA strands, which will expose the nucleotides. Now, this should be pretty obvious as to why this is the necessary first step. But anyway, it would look kind of like this. When the heat is applied, the DNA, the kinetic energy of the heat overcomes the hydrogen bonds that exist between the two DNA strands. So basically, you can picture it like this. The heat is causing the DNA to wiggle, right? It's getting more and more kinetic energy from it's thermal energy. And then pretty soon it just kind of goes like this and just separates and just kind of like, but what does not disintegrate is you don't get any disintegration of like here, right? It's only, the only disintegration you get is the hydrogen bonds, but those covalent bonds that hold the strand together, those remain intact. You'll never break those bonds. They're too strong. Okay. Now, obviously, we need to open up the DNA if we're going to start copying it. So this is the obvious first step. Now, step 2 is called annealing. And this is where we say that specific primers that we have control over, right? We add, we choose the primer. We have to pick it. So we choose it, we add it, and bind to specific complementary region, I guess you could say, on the DNA sample. And that's where the copying will begin. So what that looks like is kind of like this. So you have your DNA strands now, they're separated. And there's the primer. This is the primer, okay? The primer is going to bind to a specific region. And then the Taq polymerase will bind, and DNA synthesis will proceed in that direction. Which direction would this be? This would have to be the 3' end here, which means that this would be the 5', which means that this would be the 3'. And then that makes sense, right? Because DNA synthesis will proceed in the 5 to 3 direction. on the newly synthesized strand. Now the other side will also have a primer bind to it, and it too will proceed and create a copy. Okay, so that's step 3 is called extension. Extension is just the process when the new strand is being synthesized. So Taq polymerase binds to primer and adds nucleotides to the 3' end of the new strand. Okay, so then steps 1 through 3 are repeated 30 times to produce 1 billion copies. That's a lot. Now, the reason that this is not this is not multiplication, right? It's not like, okay, if you start with 2 strands, 2 times 30, you're going to end up with 60. That's not how it works. This is exponential growth, because the 2, if you start with 2 and then you replicate each of those, you'll have 4. And then those 4 will each replicate, and you'll have 4 times 4, and then you'll have 16, and then 16 times 16, and then whatever that is times itself. It's an exponential growth. And so that's how in just 30 cycles, you end up with 1 billion copies of that original DNA. Pretty amazing. But anyway, requires a pretty expensive machine, because the machine, again, has to very carefully control the temperatures, because there is a cooling step, and I think this is, cooling. So the annealing is the cooling phase. So anyway, any questions about that? Okay, so this leads into gel electrophoresis. So here's why. Typically, let's just take the context of the crime scene. Let's say that you did get a little hair from the crime scene. Step 1 would be this, right? Step 1 is making lots and lots of copies so that we can do as many tests as we want. So what would be the next test that we would do for the crime scene investigation? Well, now we have some suspects, right? We want to see if we can get a match. So the DNA that we just copied, we are now going to send it through gel electrophoresis and see what kind of banding pattern we get. Okay, so let's talk about that real quick. So new title is gel electrophoresis. Okay, so just looking at the title here, I'm expecting there to be some kind of electricity, because I see electro, so that's interesting. Gel, there's going to be a gel, obviously. All right, so what are the applications? It's the matching DNA samples. Okay, that's the only application. And very often that is, like we said, the crime scene, but it's also genetic relation. It could also be genetic relationships. So there's really just 2, right? It's CSI or evolutionary relationships, potentially. Those are the applications that I know of. There might be more, but those are the 2 biggest. Okay, so step 1 is there's something called a restriction enzyme. Cuts the DNA sample, cuts the DNA strand at moldable restriction sites. Okay, here's the thing. Each individual has different restriction sites. So you're going to get a different pattern. Oops, each individual has different number and location of restriction sites. So what this means is you're going to get the DNA chopped into different length pieces depending on the individual, because we all have different genetics, right? Our genetics, by the way, like if you compared any two people in this room, your genetic code would be 99.9% the same. Even like two ethnic groups, you could compare it, still 99.9% the same. But there are enough differences to create different. So let's say like individual 1. Maybe that's Mr. Roth, and then this is some student in here. If you compared the genetic, like where the restriction sites are, maybe Mr. Roth has one here, and that will get cut. Anytime I draw a line, I'm showing you where it gets cut. And then maybe Mr. Roth has one here and here. But this individual down here has one here, here, and here. And so you end up with these pieces. So imagine that these little pieces now are free. They're floating in solution. So this is a really long piece here. This is kind of a medium piece, and then 2 short pieces. This individual has really short pieces here and here, a long piece right there, right? You get the picture. So these are what we mean when we refer to DNA fragments. The fragments that we're talking about are these pieces that have been cut. So what we do then is we run them through a gel matrix, and they will separate based on their length. So we're going to add the fragments. Step 2 is, oops. Step 2 is to add the fragments to the wells. In the gel matrix. Okay, there's a couple things you need to know. One is that each fragment has exactly a -1 charge, because it's an ion. And DNA always ionizes the same way. It's something to do with the nucleotide. I think it's the OH, right? The OH on the nucleotide will ionize, and it'll have a charge of -1 because it's going to lose that hydrogen ion. Okay, so if every little fragment has a charge of -1, then therefore it experiences the same force in the electric field. We're going to induce an electric field, and each fragment will get the same force. It'll experience the same force in that electric field. We're going to apply current for 1 hour. And during that hour, the fragments are pulled pulled through the gel toward the anode. Okay, toward the anode. The anode is the term for the positive terminal. Okay, so just imagine like a battery. There's an anode and a cathode. The positive end of it is the anode. Okay, so what does this all look like? It's nice to have a visual aid. So I'm going to draw it for you. So here's the here's the gel matrix that we were talking about. Down here is the positive. You can draw positives down here if you want. And then that means that the back here is negative. And then there's these little wells, these little holes where you can inject the DNA samples. And typically you have the source right here. And then you have like maybe the suspects here. So if we're suspect samples, so maybe you have 4 different suspects and you've got a DNA sample for each suspect. The source DNA is obviously the one that was derived from the crime scene itself. And so if we see a match, then we know that we've found the suspect, right? Okay, but anyway, about the anode cathode, these little fragments are going to migrate they're going to be attracted toward the anode. And so during that 1 hour period, the fragments will migrate this direction. Now, there's a very important rule, and that is that shorter fragments have less resistance. They have the same force. All the fragments have the same force, but the shorter the fragment, the less drag, I guess you could say, the less friction or the less resistance they have in the gel. Therefore, they move further. And what that means is that so what you'll end up with is maybe like a pattern that kind of looks like this, where you've got these little bands that form. And basically, these are the longest fragments. And then the shortest fragments were the ones that got the furthest. This is making sense. Okay, that's pretty much it for biotechnology. Can everyone imagine, though? Maybe I should draw it. Imagine that you've got a banding pattern for the crime scene DNA, and what you're looking for is a match over here. And it turns out that suspect 1 didn't have any matches, but the last suspect 5, the banding pattern aligns with the source DNA banding pattern. And that's how you match it up. It's just matching. All right. Very good. We'll go ahead and end the lecture.